Hi, and welcome to BIO127 Cell Biology with Organ Systems. Today we're going to be covering just the introductory content related to this course. We will classify living systems, and in particular we will classify human individuals, human animals. We will talk about the characteristics of life, what determines if something is living or non-living, and we will discuss the levels of organization from atoms all the way up to the biome or biosphere. We'll also be discussing the scientific method, and before we start, I wanted to point out a couple of things that are going on here. So first of all, I'm a new instructor at Northwestern Connecticut Community College. Thank you for having me. So I'm taking over this course from somebody who's been teaching it for 30 years, and as such, I've decided to only change one thing at a time. I'm only changing one thing at a time, so I'm mostly keeping her content the way it is. Another thing that has happened is Northwestern has adopted a new textbook for this quarter. So a lot of the images you've seen, some of them have been updated and some of them have not been updated. Some of them are from older sources, and that's okay. They're still valid sources. I'll just be pointing out when there's something that I know about that's a little bit more up to date, or if there's something going on with an image. And I don't want you to stress out about it. I never test on a controversy. I'm never going to ask you, well, is it A or is it B? If there's a controversy of whether it is A or B, what I'm going to do is, the controversy is A versus B. Based on that, blah, blah, blah. That would be the question. So it's always going to be fair. It's always going to be reasonable, at least in my opinion. So again, I just wanted to point out some of the images are a little bit older and not strictly speaking from your textbook. Classifying living organisms is not strictly from your textbook. Characteristics of life is, I don't think, strictly textbook content. Maybe it is. And then the scientific method is actually not in this chapter 1 of your textbook either. That having been said, it's definitely worth going over. These are very fundamental, and this is a Gen Bio with systems or overview content. So that general biology perspective is about being a scientist and understanding what it is to think scientifically. And this is really the starting point for that. You also may be wondering what's up with this glove right here, because I wave my hands around a lot. And I have a drawing tablet. I may or may not use it right now, but this helps protect the drawing tablet from my hand and allows me to draw on it while scraping my hand all over it. That's what that's for, so that's just, don't worry about that. Okay, so we need to move on to how we're going to classify organisms. There is a whole phylogenetic tree, and we're not going to do the whole phylogenetic tree. It starts with what domain of life an organism is in, and there's 3 basic domains of life. Humans are eukaryotes. That means that the cells in a human body have a true nucleus, lots of membrane-bound organelles. And the best way to differentiate that from the other domains of life is to point out that bacteria and archaeobacteria do not have true nuclei. They do not have membrane-bound organelles. All of the contents of the cell are just kind of loose within the cell. It's less differentiated, it's less specialized. And that's really it. I don't want to spend too much time in bacteria or archaea. And in fact, the last time I talked to a microbiologist, she was ranting about how the entire domain of archaea is being rethought. We used to think archaea was like these extremophiles. They only exist in volcanoes or in the near deep sea vents, and that's proven to not be really correct. And they really seem to be a lot closer to prokaryotes than we previously thought. We're finding them all over, and the differences between them, they're just an overhaul. It's in flux. So we're really focusing on human biology in this course, so let's focus in on that eukaryote domain. So we're in eukaryote domain along with plantae, fungi, and protista, which is to say plants, mushrooms, other fungal things. Those are actually relatively closely related to us compared to bacteria. Now again, eubacteria are single-celled prokaryotes, so they are not really going to be in our domain, but the rest of these are in our domain. We do have We are the kingdom Animalia. Humans are kingdom Animalia, so that's the one you're focusing in on. We are heterotrophs, and I'm a big proponent of linguistics. I want you to be learning medical language while you are learning scientific language and learning your cell biology and anatomy. So if you're studying any medical language as you go along with this, you're going to be able to break this word down. And eventually you'll learn that hetero simply means different from. And troph indicates some kind of eating. So heterotroph means it eats something else. So the significance here is that animals eat something else to gain energy for their own cells. Now, why this makes us different from other kingdoms, if you come down here to kingdom Plantae, they're still eukaryotic like we are, but they are photosynthetic. So we're getting energy from things that we ate outside of ourselves. We're taking other cells and giving them to ourselves, and that gives us energy. Whereas a plant is able to photosynthesize using light. So that means it's getting its energy from within. So you wouldn't use heterotroph for that. It's photosynthetic instead. Let's see, fungi, the kingdom fungi, those are still eukaryotic as well. Fungi, yeast, mushrooms, those are all examples of the kingdom fungi, and they have a role in the biome as decomposers. They're part of the nitrogenous waste cycle. They're doing a lot of the dirty work and recycling a lot of really important nutrients and then giving it back to us. So they're really, really important and useful in the cycle of life. And then there's the kingdom Protista. They are eukaryotic protozoa. That could be algae, it could be slime molds, really, really simple organisms that still have the true nuclei, still more complex than you would see in other domains. Now, I was wondering if this was out of date. I can't find anything more up to date, despite my conversations with microbiologists ranting about what archaea actually is not. So again, because of this controversy, let's focus on eukaryotes. Let me go this way so I'm parallel. Let's focus on eukaryotes. Let's look over in this corner where we have all of our true nuclei, cellular, multicellular organisms, or single-cellular organisms. And here we are right next to fungi. It's hard to tell from the way this chart is arranged, but we are actually more closely related to fungi than we are to plants. We actually have a lot in common with fungi at the cellular level than plants. And that's really where I want us to go with this. That's really it. This is a human biology focused course, so let's stay over on this side of this chart. Now I put things a little bit out of order. I'm already talking about cells. Cells are such a fundamental thing. We're going to go from atoms to molecules to cells. We're going to spend a considerable amount of time talking about cells, thinking about cells, considering the significance of cells. I've already started using terminology related to cells, like how they have nuclei if it's a eukaryotic organism, which we are, all of the organelles. That's all going to be really important vocabulary, so if you are not strong on that vocabulary, you might want to read ahead, or you might want to, think back on your high school biology experience. Those terms are commonly utilized. We're going to keep using them. So the characteristics of life that we're looking at right here. This is how we determine if something is alive or not. And it's actually more difficult than you would think. It's not as easy as, it's moving around, so it's alive. Robots can move around. Are they alive? It's not as easy as, being able to complete a task. Same deal. Some kind of robot, some kind of computers, those are kind of complete tasks. So we actually have to have some pretty sharp definitions of what is alive and what is not. And it turns out that there are some controversies in here. There are some things that straddle the line between alive and not alive. Do we consider a virus a living thing? Now, in order to figure out if a virus is a living thing, we need to understand what a virus is. So first and foremost, let's look at the characteristics of things that are alive. And I reordered this. Whoops, didn't mean to do that. I meant to highlight. There Living things are composed of cells. Now, again, a cell is made up of several things. It's going to have a plasma membrane. It's going to have organelles. It's going to be made of the 4 macromolecules of life, which we're going to be getting to as we move past our chemistry in the first part of this course. So it's made out of 4 fundamental things: proteins, nucleic acids, carbohydrates, and lipids, fatty acids, fats. So 4 things, that's what you're made out of, plus vitamins and minerals. Every cell in your body is made of those 4 things. Your hair, your eyeballs, your teeth, 4 things, plus vitamins and minerals. So in order to be considered a cell, it has to have all 4 of those things. A virus typically has 2 of those things. A virus typically is made out of nucleic acids and a protein sheath. So if it doesn't have those 2 other components of the cell, it's not technically a cell. And if it's not technically a cell, then it's not technically alive. So let's go ahead and look at the rest of these things and then come back to the idea of that virus and ask ourselves if it's alive. Requires energy and raw materials. In a way, yes, a viral life cycle is kind of parasitic. Now, if you're talking about human cells, absolutely. We require energy and raw materials. We eat a lot of food every single day. On average, between males and females, that's a 2,000 calorie diet per day. For females, it's closer to 1,800. For males, it's closer to 2,200. So 2,000 doesn't work for anybody. And of course, everybody has individual variation between those numbers. So we have all these nutrient needs. We require energy for raw materials. Yeah, okay, that works for we're alive. We maintain homeostasis. We will be talking about homeostasis shortly. It is in this introductory chapter. Homeostasis means that we're not going to let our internal environment get too far away from our normal. We have body temperature. We want it to be about 98 degrees. We don't want it to get to 100 degrees. We also don't want it to get to 95 degrees. Both of those are very, very dangerous situations for the cells in our body. If they get too hot, we cook our cells, and they start to die. If it gets too cold, then we freeze our cells and they start to die. So that's a very serious thing, and that's a homeostatic mechanism. So we have all kinds of homeostatic mechanisms to keep us in that happy midline. Response to the environment. Humans definitely do that. So if it's cold outside, we'll put on a coat. If we're thirsty, we drink water. Don't just think about humans. There are single-celled organisms. The most fundamental unit of life is a single-celled organism. They do these things too. They also require energy and raw materials. They also maintain homeostasis within the context of that single cell. They also can respond to the environment one way or the other. Usually it's chemical if you're talking about a single-celled organism. So responding to the environment as a human is putting a coat on. Responding to the environment as a single-celled organism is probably about chemoattraction to a different area. Growing and reproducing, if you're a single-celled organism, there's probably some kind of like splitting into 2 that you're going to do, and then there's 2 of you. If you're a human, you're going to produce gametes, and reproduction is going to happen when you join up gametes with other gametes, and are capable of evolving. This is obviously true for humans. Now, evolution, we can talk more about evolution. The way I always frame it is, you don't have to believe it, you do have to learn it. As a scientist, you have to learn and understand what evolution is, and that is simply that organisms change over time. And the fact is they do. We can see it very, very easily. If you have a purebred dog, then you have seen forced evolution. If you've ever had a strain of antibiotic-resistant bacteria, then you have seen evolution in action. Evolution is when DNA changes over time, and DNA changes the way physical attributes look over time in very, very subtle ways. And if that change in the organism is beneficial, it makes them reproduce more, then that change to the DNA is more common in a population. And that is its whole lecture. That's a whole course. We could spend all day on that. But I just want you to know where I stand on it. It's for real. If you don't believe in it, fine, just learn it anyway. So every organism that is alive has an evolutionary history that we can trace back. And we've already seen what that evolutionary history looks like. We are Animalia, we are eukaryotes. We evolved from the most primitive eukaryotic organisms. Now, viruses are capable of evolving too. They do have nucleic acids, they do have a genetic code, therefore they do evolve. So, do you think a virus is alive? Based on what you know about viruses, you can even read up a little bit more about viruses. Think it over, decide for yourself. There isn't a proper answer for that, actually, no matter what. I could have you submit your answers, I could have you debate it in a discussion forum, but there's no right answer here. It's the grayest area in this field. Now, this course being Cell Bio and Systems Overview, we are going to be looking at the levels of biological organization. We're going from atoms to organism. And really, it's going to be an organ system basis. So we'll start with atoms and molecules very, very soon. We'll move on to several weeks of cell biology. So you start binding your atoms together. You bind enough atoms together, you have a molecule. If molecules start arranging themselves in a complex way, you start to have a cell. If cells are arranged together and they ultimately have a common function, you have yourself a tissue. That tissue has a function. You put multiple tissue types together that serve a common function, you have an organ. If you have several organs in a grouping with a common function, you have an organ system. And if you have multiple organ systems, you have an organism. So, human. Human is an example of organism. And that's as far as this course goes. This is from your textbook, and for whatever reason we've cut out the organism here. It's an image of an infant in your textbook. So again, atoms bind together to make molecules. Molecules bind together to make tissues. Sorry about that. Tissues bound together, molecules bind together to make cells. Cells accumulate together to become a tissue. Tissues accumulate to become an organ, and organs connect to each other to become an organ system. So when we say systems overview, we mean that towards the end of this quarter, second half of this quarter or so, we will just do a broad overview of every single system in the human body. And that's going to set you up for success in your anatomy and physiology courses of the future, much more than a Gen Bio course would. Now, just to point out, again, we're starting at atoms and molecules and ending at organism. If you were to take a Gen Bio course, like maybe Gen Bio 2, or maybe move on into like an ecology course, you would probably move on to studying populations, communities, ecosystems, and biospheres. And these essentially work the same way as we have all the way since atoms. You take multiple organisms that have commonalities, now you have a population. You take multiple populations, now you have a community. If you take multiple communities and all of them plants and non-living things in an area, you have an ecosystem. And if you take all of the ecosystems on Earth, or all of the ecosystems together, you have a biome, and ultimately a biosphere. So this is what you have to look forward to if you start to go more of the general biology pathway. Now, the last thing to talk about in just our introductory part of this lecture is being a scientist. Just the very foundational, what is a scientist? How do scientists think? How do scientists report? There are two major ways to reason. We can do inductive reasoning, and that's the kind of Sherlock Holmes type of reasoning. It's like, oh, he was wearing a yellow suit, therefore he was the murderer. You take one little factoid and you extrapolate. You say, if it's true for this little thing, it's true for all of these things. it's not always perfectly accurate to say it's true for this, then it's true for everything. If it's true for this mouse, then it's true for humans. We do have science based on this, right? We do research on mice, and we start to base medicine and do human testing based on the evidence in mice. So from specific to general. The cells in the mouse are going to act more or less like the cells in the human. Deductive reasoning is from general to specific. If it's true for everybody, then it's true for this individual, which is pretty solid reasoning usually, but we have statistical variation. So neither form of this is perfect. In order to get to the most ideal understanding, we've developed the scientific method. How do we go from not knowing something to having some evidence for that thing? Now, this doesn't mean I've done the scientific method, I am now correct, I'm done, go wash my hands of this, and I go home. No, what this means is you have evidence for a thing. So what you do is you make an observation. You have all of the science that's ever existed in the history of mankind and the scientific method, and you look at all of that evidence, and you formulate a hypothesis based on it. So you say, you know what, let's apply this to the light in your refrigerator, right? Is the light in your refrigerator always on, or does it turn off when you close the door? How do we know? So we can accumulate data. We can stand at the fridge and open it and close it and open it and close it and open it and close it, and we make observations based on that. And the light's always on when we're looking at it. So maybe you formulate the hypothesis that the light is always on. So you can make some experiments based on this observation. So maybe you want to say, I'm going to put a camera or a video recorder. How about this? We're going to put a video recorder inside of a refrigerator. And then it's going to tell us if the light is on or off when we close the door. So you put the video camera in your fridge, you do your experiment, you close the door. Maybe you do it several times because repetition is important in the sciences. You open the door, you close the door. You open the door, you close the door. If you're really good at science, you're also recording the time that all of this occurred. I opened the door at 10:57 a.m. I closed the door at 10:58 a.m. I opened the door at 10:59 a.m. And so on and so forth. And then you can match that to the timestamp on your camera. And because you wrote it down, because you were very systematic about it, your evidence is much stronger. So now you have new data. And, well, your new data says that the light is off when you close the door. So that contradicts your initial hypothesis. So you're going to want to revise your hypothesis. And if you're really doing things correctly, you'll revise your hypothesis. The light turns off in the refrigerator. And you'll redo the experiment. And this is going to give you new data that supports your conclusion. And if you repeat this enough times with enough scientific validity, maybe somebody else comes along and says, I don't trust your video camera. I'm going to put this photon sensor into the refrigerator. And that's going to give us better data because that's photons. That's not, maybe it's cold in there and it's shutting off your camera. You don't know, right? There are all these ways that other scientists are going to nitpick your data, and they then are responsible for responding with better modalities to record that information. So you test and you test and you test, and every time you test and you get the same conclusion, you have support for your hypothesis. If something gets supported over and over and over and over and over again, then we can call it a theory. If it's never been contradicted, then you have a theory. And that's what evolution is. Biology, the field of biology, does not have laws. Physics gets the laws. General biology gets theories. So it's kind of a linguistic problem that we have in communicating with the general public when we say evolution is a theory. What you're seeing from the scientific method is we've tested it over and over and over and over and over again, and our hypothesis is always supported. Our hypothesis that evolution is real, evolution happens, it is occurring right now, at the micro level, at the macro level, it is occurring all of the time. That gets supported every time we test it. So for that, we get the theory of evolution, and that is the highest you can get to in the scientific method. There is nothing over theory in the biological sciences with the scientific method. That's the peak. That's the most you can hope for. Okay, and then if you're really, really smart, you don't just say, okay, we know that the light turns off in the fridge when the door is closed. Maybe you go about looking for the button that turns it off, and then you do a bunch of experiments based on that button that turns the light off. And that's when people get PhDs, is when they take the old evidence and they take it in a new direction. So again, the important thing here is observations based on all scientific information, formulate a hypothesis, create an experimental design, develop new data. The data either supports or refutes your observation. Based on your data, you develop a conclusion. If enough data accumulates that always supports the same conclusion, then we can eventually form a theory. One thing we don't do in science is try to prove ourselves correct. So that's very careful wording. We don't do science to prove ourselves correct. We do science to become more correct. We go from not knowing to having some evidence, right? That's what we're doing. So, as an example of this, when I teach my pathophysiology courses, we do something called differential diagnosis activities. And when a patient comes in and they're just clutching their liver, you don't know what's wrong with them yet. There are about 50 things that could be, 100 things, 1,000 things that could be causing them to clutch their side like that. Maybe they got stabbed, maybe they have hepatitis. You don't know. It's a differential. It's different. There are different possibilities. But one thing I see my students do is they get caught on an idea. They say, I think that this person has been stabbed. And that's the only thing they put down for the differential. They got stabbed. I'm going to check to see if they got stabbed. This is a really bad example. They don't really do that. They do something else on this particular, but whatever. So they get stuck in this idea. And then the only test they do is a visual assessment to see if that person got stabbed. Now in this case, it's not that they got stabbed, it's that they had hepatitis. So the same is true for science. You know, you don't decide ahead of time that the light is off in the fridge. You make an observation, you make some assumptions, and you test it. And then if you are incorrect, that is still an advancement. That is still more data than we had before. So the null hypothesis, proving yourself incorrect, is just as important as proving yourself correct. And that is true for the scientific method. And the scientific method is the basis of medicine as well. So just like on that differential diagnosis activity, we don't think of one diagnosis and then test for that one diagnosis. We think of multiple diagnoses. We test to rule some of them out. So it's very similar to what we're doing with the scientific method. I just said all that, but it's nicely written down for you. Now that you have data, you've accumulated enough data, you've run multiple experiments, what you can do, the best outcome for you, is to write it down and have it peer-reviewed. That means you're going to formally write out your results. You're going to write out your experimental design, you're going to write out your results, you're going to discuss your results and what you think they mean. And before it gets published, it gets sent to at least 2 other experts in your field. So going back to the refrigerator example, there are 2 refrigeration experts, and they're going to read your evidence. And either they're going to like your research or they're going to hate your research. One of them might say, whoever thought of putting a video camera in a refrigerator? That's poppycock. And they might send it back to you, and you might have to make revisions before that gets published. So it's very rigorous. And not every finding will get published because it's rigorous. That means if it's in a peer-reviewed journal, you know that the leading experts have looked at it and read it and possibly recommended revisions, and that That is as good a paper as you can get in the sciences. That is as accurate as it gets. It's still not perfect. Humans aren't perfect. The better you get at science, the more of those papers you'll read, the more problems you'll find in them. But it's still the best we have for science-to-scientist communication. Now, of course, it's at a very high level scientifically. We're not really talking about refrigerator lights. We're talking about phosphokinase pathways. We're talking about research in medicine. We're talking about, details of metabotropic pathways. So the general public is probably not going to find it accessible. They're probably not going to be able to use that information. Now, an intermediary to the public might be the popular press, somebody who is literate scientifically, but hopefully has some training in communicating with the general public. So they might read your new paper on your miracle cancer cure, and they might translate it into something the general population can read and understand. If they're doing well, if they are good at their job, then they can communicate that successfully to the general population. Unfortunately, the popular press is not always very good at communicating findings to the general population, and you get all of these clickbaity headlines like chocolate is the cure for cancer or chocolate causes cancer. Neither of those is accurate, but it gets people to read their article. Some are better than others, though, and I have read some very good popular press articles that fairly well summarize the papers that they're trying to present. If you're a scientist, you can find things through the popular press that you're interested in, but you always want to click through to the peer-reviewed journal and read that for yourself and assess the quality for yourself. And then electronic publications, it's highly variable. You can have really good online things like the NCBI is very, very good. I mean, they have a lot of peer-reviewed journal on it anyway. There are some good online sources, but anybody can put anything online, so you do end up with problematic online publications. Your Auntie Karen can definitely go on online and write a publication about how face masks are causing pneumonia, and I promise you face masks are not causing pneumonia. And if you're curious about why face masks do not cause problems with carbon dioxide, or how they do not cause problems with pneumonia or low oxygen saturation, I am happy to tell you exactly why none of those are problems. But that just goes to show you how problematic electronic publications can be. We've already talked about what a theory is, so I won't review that. I do want to sort of do a better scientific method for you. So I did refrigerators, but a better experimental design, or a good reason to have an experiment, might be to determine if a certain substance is carcinogenic. And how might we design that experiment in a way that gives us good evidence for our hypothesis? So let's say a new artificial sweetener came onto the market, and we happen to notice that there were more cases of bladder cancer. My cat has decided to become part of this cast today, so I'm going to pull that and pull his butt. She's a cat, so of course her butt's here. So, okay, correlative. That correlates. There was a sweetener on the market, and more people started getting bladder cancer. Oops, that's a problem. But correlation is not causation. Two things that happen at the same time in no way mean that one caused the other. So we're going to make a hypothesis based on observation, more artificial sweetener, more bladder cancer. And we're going to run an experiment about it. And we're going to make an ethical experiment that does not harm people, at the very least. It's going to have to get through ethics review boards, IRBs, internal review. It's going to have to get through a lot of people before you can do this experiment either. And you're going to have to describe your experiment in detail in advance. So you're going to get, let's say, 50 mice. And you're going to split them into two groups, an experimental group and a control group. And you always want a control group. In this case, you want a control group in case, well, what if these particular mice, just have an increased rate of bladder cancer? That's going to mess up our results. So you're going to have a control group just in case there's something wrong with the mice to start with. Control group gets none of the sweetener. They get a standard mouse diet. You're going to keep them in good health. Everybody's going to be happy and the same. In the experimental group, everything's going to be identical except that they additionally get the sweetener in their food. A correct amount of time passes, maybe, let's say, a year, enough time for bladder cancer to establish itself. I'm making that number up. I don't know how long it takes for a year for bladder cancer to establish itself. And let's say after that year, you examine all of the mice in both groups for bladder cancer. Maybe it's just palpative. Maybe you have to do some surgery, remove their bladders, killing them. Sorry, mice. It's for the sake of humans. It's up to your experimental design. You get to decide. I'm not deciding this for you. If your experimental group has significantly higher rates of bladder cancer than your control group, now you have evidence that your sweetener might be causing the bladder cancer. But again, this goes to peer review. Maybe it gets published, maybe it gets criticized. And if it's criticized, they might look into your methods. Maybe you accidentally made something else different about the two environments. Maybe the amount of sugar you gave them was ridiculously high. No human is going to have that much sugar in their diet, which humans are going to have that much sugar in their diet. But all of these criticisms always come through. And so you're going to want to test this again and again and again. The more organisms you can test this on, the more times you can repeat this experiment with the same or similar results, the more evidence you have for your hypothesis. You present your data on graphs and charts. You use numbers. You do a statistical analysis. So we could even do multiple experiments with increasing amounts of sweetener in the food. We can go from 0 sweetener, 0 cases of bladder cancer. Whereas if you have, 20, this is not a good graph because this needs values. Let's say 20 milligrams of sweetener in their food every day. 20 milligrams. If you have 20 milligrams of sweetener in your food every day, you get approximately 10% chance of having bladder cancer, which you've increased your value tenfold. Let's say this is 1. So now it's at 10. And if you have 30 milligrams of artificial sweetener in your food, now you have a 30% chance of developing bladder cancer. And this would have to be consistent. This would have to be daily. Oh, it's a percentage, isn't it? Oh, sorry. They do have values. I don't see that. That's a good value, though. I would prefer milligrams for that. So scientists are really picky. If you see me being picky about this chart, it's because scientists have to be very, very picky, because other scientists are going to look at this and say, why is that a percent? The percent of your food that is sweetener? Nobody has 30% of their food as sweetener. It's just like you're eating hard candy all day. That's disgusting, but we're Americans, so we do it. Here's another example for Lyme disease, how we made observation, a hypothesis. We did experiments, and we got results. This being Connecticut, or you being in Connecticut at the time of this recording. At the time of this recording, I'm in Denver. Connecticut is where we discovered Lyme disease. It wasn't even actually scientists who discovered Lyme disease, as much as this is a useful way of understanding the scientific method. It was actually a mother of several boys. I'm going to say it's in the 1950s, but I'm not very good with years, so don't count me on that. She actually kept taking her kids to the doctor. They kept getting all of these, rashes shaped like a dartboard. What's it called? Bullseye. There we go. Bullseye-shaped rashes. And they always had joint pain. So her kids were out running out in their fields in rural Connecticut, and they kept coming home with all these rashes and joint pain and fevers and problems with cognition. And the doctors didn't believe her, and she started recording literally everything about what her kids were up to, what her husband was up to. And after showing a physician her notes, then they started doing these experiments. So that was actually science that was initiated by the general population. That was not scientist-initiated research. And that's important, too. It shows you the importance of recording your information. Nobody's going to believe a word you say unless it's written down. So, of course, this mother of several children in rural Connecticut started thinking about the horrible conditions that her kids were going through on a regular basis, she was skeptical of her physician. She was skeptical of her primary care physician and his insistence that there's no disease that causes this. This isn't born. This has nothing to do with environment. She was skeptical of that. She wrote everything down. She wrote down how they got ticks, and then there was a rash around the site of the tick, and then how there was a different time frame that all of these different symptoms developed. She wrote it all down, so she appreciated the value of statistics. She probably didn't do the graphs. The scientists probably did the graphs. We also have to distinguish anecdote from scientific evidence. The doctor heard of one woman whose family was getting sick. Maybe it's genetic, right? You have to think very carefully. So there's a good reason for that physician to tell her, no, there's nothing wrong with you. This is an anecdote. It is not scientific evidence yet. You have to separate your experimental results from your conclusions, right? Just because you got evidence that the light turns off in the refrigerator does not mean for sure that the light turns off in the refrigerator. Your results support a conclusion. They do not confirm a conclusion. So a lot of people are going to have to nitpick and ask you, are your conclusions justified on the basis of your experimental design and results? And again, understand the difference between correlation and causation. So there are big problems with correlation and causation that cause a lot of controversies in our society. So for example, we had increased rate of autism spectrum disorder diagnoses, and we had an increased rate of vaccination. Does that mean that vaccination caused autism? I'm really hitting all of the controversy notes today. I'm probably alienating like half of you right now. I really hope I'm not. And if I am alienating you, I'd much rather have a conversation with you. So please do book office hours with me if you are an anti-vaxxer. I'd really like to walk you through the evidence and have you be a critical thinker and have you be a scientist about what the evidence actually is. That's what I'm aiming for. But because these two things happened at the same time, more diagnoses, more vaccinations, a lot of people came to the conclusion that vaccinations cause autism. But when we do the research, when we set up valid scientific experiments, that hypothesis is not supported. And now you understand what that means is that there is no evidence that vaccines cause autism. Even when anti-vaxxers fund research into vaccines causing autism, they come up with the results that no, there's no connection between vaccines and autism. And I can prove that with science. We have proven that with over 500,000 individuals, millions of dollars in scientific funding. More in funding for research based on the claims of anti-vaxxers than any kind of profits that pharmaceutical companies are making on actual vaccinations. So, again, if you're an anti-vaxxer, I'd rather have a conversation with you. My personal belief is that if you alienate anti-vaxxers, they stop getting health care. And if they stop getting health care, that's very, very bad for their children, and it's very, very bad for them and their own health. So my goal is not to alienate. My goal is to invite you into the party of being a scientist and being a critical thinker. Science improves technology and the human condition. Science has limits, as we already saw. Up until the 1950s or so, we didn't know that Lyme disease existed. It really did take individuals coming forward with serious problems to convince scientists to actually start studying this thing that was happening in a very specific region. But eventually scientists listened. Science takes time. Science has to be repeated. So by the time science is actually saying, yes, there's Lyme disease, a lot of people have already been affected. So science does have its limits. A lot of them are time limits. A lot of them are based on how nitpicky scientists are. But if we weren't nitpicky, then we would never be able to prove something, right? So all of this science allows us to make informed choices about using the correct types of bug spray, something that's going to adequately keep ticks away or keep mosquitoes away, keep all of those diseases that are spread by those organisms away from you. I've already bought this because I'm moving in like 2 weeks and I want to be able to put it on my dogs. This is specifically for dogs. right when I get there, because I really don't want my dogs to get Lyme disease, and I know they're going to bolt off into the forest right when I get there. So science allowed me to make an informed choice. Knowing about the prevalence of Lyme disease in Connecticut helped me make a choice about the health of my dogs and myself. And that's it. I've talked your ear off for 45 minutes, and please proceed to our Chapter 2 content.